today's episode of Myeloma Crowd Radio, a show that connects patients with myeloma researchers. I'm your host, Jenny Ahlstrom, and we'd like to thank our episode sponsor, Takeda Oncology, for their support of Myeloma Crowd Radio. Our show today is about allogeneic or donor stem cell transplants and how to use this type of transplant, which is actually the very first immunotherapy ever used in myeloma. Dr. William Matsui from Johns Hopkins has made significant progress making this type of transplant safer and is working on making it more effective, and his research also focuses on the early precursor cells, so we'll be covering that as well. I just want to note before we get started, if you'd like to meet Dr. Matsui in person, we welcome you to join us at the upcoming Myeloma Crowd Radio Roundtable on Saturday, March 24th, where Dr. Matsui and several other myeloma experts will be joining us to talk about the latest research for relapsed myeloma, high-risk myeloma, and myeloma innovation. You can join this and 3 other roundtables this spring, so you can check out our website at myelomacrowd.org to find one near you. So, Dr. Matsui, welcome to the show.
Thank you so much for having me.
Okay, we're so happy you're here. Let me introduce you before we get going into our questions. William Matsui is the head of the myeloma program at Johns Hopkins University. He's also professor of oncology at the Sidney Kimmel Comprehensive Cancer Center and is a member of the Department of Oncology and a member of the graduate program in pathobiology and cellular and molecular medicine. He's on the editorial and review boards of Hematologica, Immunology, and Immunogenic Insights, is a SPORE recipient award researcher in lymphoma, and has won multiple teaching awards in the Johns Hopkins Department of Medicine and Oncology. He's received the Kimmel Foundation Scholar Sidney Kimmel Foundation for Cancer Research Award, as well as the LLS Scholar in Clinical Research Award. Presently and for the last few years, Dr. Matsui has served on the NCI Investigational Drug Steering Committee and is the co-chair for the Cancer Stem Cell Task Force. And just as I mentioned, Dr. Matsui is moving west in the next few months and will be in Austin, Texas in June at the Livestrong Cancer Institute, a brand new school, part of the Dell Medical School at the University of Texas in Austin. So I know patients that are a little further west will be very, very excited to hear about that. And again, welcome to the show.
Thank you.
Well, Dr. Matsui, why don't we start by talking about allogeneic transplant as an immunotherapy, because right now we hear about all these different immunotherapies coming out, and that was really the original.
Yes, so allotransplantation was actually, like all transplantation, was never, originally meant to be an immunotherapy. All transplantation, including autologous transplantation, is the best example of this. Using stem cells from yourself or someone else was really meant to be a way of overcoming the bone marrow toxic effects of high-dose chemotherapy. And so when people were doing this way back when, they wanted to, the thought was is that they wanted to use blood or bone marrow from somebody other than the patient. And the point was is that person had no leukemia or cancer, and so you weren't transferring that back into the person. So as a side effect of that, people would have these immune reactions where the immune system of the donor would start to attack them. And what people later figured out was that actually, those effects were actually beneficial in terms of controlling cancer. And so there's been sort of a lot of, questions about, well, does the immune system really play a part in controlling cancer? How does it do that? How does it work? And I think that, in the case of allogeneic transplant and myeloma, the best example that is immunotherapy is that if you have a transplant and then you take more immune cells from the donor, if the patient is unfortunately relapsed after the transplant, you can give them those immune cells and you can see the tumors go away, actually. So that's really, in part the evidence that it is an immunotherapy. And since we've been doing bone marrow transplants for, almost easily 4 to 5 decades now, it's probably the oldest sort of established form of immunotherapy. and it is it's been used in all different heme malignancies, and it has had a checkered past in myeloma. And I think that, at this point in time, maybe there have been enough advancements that we can actually go back and take another look at whether or not it might be a useful thing to add to the armamentarium of things we do for myeloma patients.
Mm-hmm. Well, let's talk about that. So why allo in myeloma and why not allogeneic transplant in myeloma? What would your take be on that?
So the reason why I think that allogeneic transplantation should be considered is that if you look at long-term studies, and these have been studies where people have been followed for over a decade, for sure there are people who have undergone transplants and have no evidence of their disease coming back. And that, coupled with this idea that it's immunotherapy, I think makes people believe that some portion of patients, for sure not every patient, but some portion of a group of patients is potentially cured by this approach. And that's the same in leukemia. It's the same in lymphoma. We use allotransplant in those diseases because we want to cure the patient. So that's for sure the good part about it, right? Like potentially curing someone is definitely a good thing. The downside has been the side effects of it is number one, and then the other thing I would say is that in this day and age, where you have many different therapies for myeloma and patients obviously are living longer, is it better to just keep treating with a bunch of different things versus undergoing something like a transplant in general? So I think that on the side effect side, it got a very bad rap in the 90s, and people started doing transplants, allogeneic transplants, sort of in a very concerted fashion in the mid to early 90s. And so when I was doing my medical training, I went to medical school at UCSF, but I did my internship and my residency at the University of Washington in Seattle, and that's where the Fred Hutch is. They were doing a lot of transplants specifically for myeloma at that point in time because myeloma is a very common disease compared to a lot of other heme malignancies. And the problem was that at that time, there was almost a 50-50 chance that you would potentially die from the procedure, right?
Wow.
So that is super high. That is for sure unacceptably high, right? Especially for some disease that can be a chronic disease like myeloma. You don't want to provide a 50-50 chance of not making it, 6 months.
Through the procedure, right.
Yes, exactly. Right. And so that's why it was very much sort of downplayed. And so one thing to think about is that, well, why was it that, myeloma patients became so sick from that procedure, and are those things that you could fix over time? So really there are two parts to, there are two major issues in myeloma patients. One is that if you give patients, for whatever reason, high-dose chemotherapy in the context of an allotransplant, and this is probably a little more chemotherapy than you would get, intensity-wise, than you would get with melphalan, just giving 2 days of melphalan. And with that, it was difficult. There was a lot of organ damage that was done. People had a lot of pneumonias. There was renal problems. Giving those higher dose of chemotherapy to people. And then the second issue is that even if you survived the chemotherapy part, then you have this immune system, and the question is that, can the immune system tell the difference between you and your myeloma? Because if it can go after the myeloma, then that's all good. If it now starts to attack you, that's something called graft versus host disease. You never see that in an autologous transplant because the immune system that you're getting back in an autologous is your own, so you're not going to recognize yourself as being foreign. But if you're bringing in a new immune system, it can start to attack your organs, and the most common ones are your skin and your liver and your gut. And if there's a very profound or intense reaction, then you can die from that immune reaction itself. So back in the day, we were straddled with this conundrum of, patients, we could potentially cure some of them, but, we were probably hurting more people than we were helping at that point in time. So I think there have been some major advancements in the last 30 years that have now allowed us to make the transplants, an allo transplant, much, much safer. And I think that if you get rid of, this crazy nature of having, every other person not make it through the transplant, then I think that now you can go back and consider it. But like if you talk to most people who are myeloma folks, they have one point or another tried allotransplantation, and it is more technically complex than doing an auto transplant. And there are many more complications from doing allotransplantation. So most people don't like doing them.
Right.
And I'm a bone marrow transplanter, and I transplant all diseases, including myeloma. And so those things that we see in allotransplantation for sure are more than in an auto, but those are complications you see for every disease. And so I think that to me is not as big a deal. And so I think that when you bring up the question of allo with most myeloma experts, I think that it's, like I said, it's interesting to hear the spectrum of sort of comments you get back.
Right.
Like it's too dangerous.
I'm get a very wide variety.
Some people are still, for them, and are still willing to try them. Other people say, that is like so dangerous, and half the people die, and then the other half get graft-versus-host disease. And like I said, I think those are individuals who don't necessarily do allogeneic transplantation. If they're really only taking care of myeloma patients, they're probably only doing autos. So there have been these incredible, I think, changes over time. There's two major ones. One is that instead of using high doses of chemotherapy, which is what we used to do, we used to give 8 days of high-dose chemotherapy or 4 days of chemotherapy and 4 days of radiation. You can actually get by with very low doses of chemotherapy, and it's very similar to the chemotherapy that you might get before you get CAR T cells. So that chemotherapy is really not designed to kill the myeloma. Its job is to try to sort of trim back the normal immune system so that, like in the case of CAR T cells, you're making space so the CAR T cells you've constructed can go into your body and now have room to expand. For us in allotransplantation, we want to make the patient's immune system as stupid as possible so that what we can do is put someone else's bone marrow and immune system in there, and it won't get automatically rejected. And those medications, there are two in particular. One is cyclophosphamide or Cytoxan. That's used sometimes in myeloma. The other is called fludarabine, which is never used in myeloma. But those two together are very potent immunosuppressants. They're not necessarily things that completely kill your bone marrow, but they're really designed to modulate the immune system and get rid of the immune system in the host. So once you do that, then it becomes pretty easy to put another person's marrow in. And so the big advancement about 2 decades ago was that you could use low doses of chemo rather than high doses of chemo. And with that, over time, it's become gentle enough that, when I started doing transplants, the upper age limit to have an allo transplant was usually in your mid-50s. And now it's well tolerated enough that here at Hopkins, we have no upper age limit, actually.
Wow, that's incredible.
Yeah, so And the other thing is that, it used to be when you did a bone marrow transplant, you would be in the hospital for easily about 7 or 8 weeks, right? And for us now, we do, here at Hopkins, we do like 90% of them as outpatients. And, the vast majority of people never end up being an inpatient. So it's mild enough that people are able to get up, walk around, go back and forth. And so we have a clinic, and they come back and forth to that outpatient clinic every day while they're getting their transplant. But it doesn't mean that they need to be cooped up in a hospital for, 40 days and 40 nights anymore. So it has gotten much, much better in that way. And then the second thing is this issue of graft versus host disease, because that by itself is a major side effect. Like I said, you never see that with an autologous transplant, but It can be severe, and there's been a lot of efforts over the years to actually decrease the amount of graft versus host disease that you can get. And here at Hopkins, we have a specific way of doing it where we use cyclophosphamide again, but we do it after you get your transplant. And if we do that, we can take, let's say in the past there used to be graft versus host disease rates of about, serious graft versus host disease rates of about 50%. And now we've reduced those to, probably easily less than 10% and probably more like 5%. So if we can make it safer, then I think it now opens up the possibility to see whether or not, how curative is it, and are there strategies actually that you can try to think about to make it more effective over time. But I think that, it's hard to, if you think about it, it's hard to make things a whole lot safer and make them a whole lot more effective at the same time. It's almost the opposite effect. If you make it more safe, it doesn't work as well.
Not as a,
but,
right.
But I think that since we've vastly improved the safety, now it's time to really work on the efficacy. So a lot of the trials that we do here at Hopkins have been really focused on, well, how do we make sure that people don't relapse as quickly or relapse, following the allo transplant. And, having a new immune system, I think, provides a very good platform to now do a lot of things, including trying to harness that donated immune system against myeloma.
Well, that's just totally incredible. I didn't know that the low dose chemo was what you're using to do the actual transplant. So do you lose your hair and things like that if it's such a low dose? I mean, you're still taking the chemo.
Yeah, so it's a little bit different. So, like, I think that if you've experienced an autologous transplant, you probably, with melphalan, you probably lose your hair. You can have issues with nausea. You can have issues with mucositis, where you get, mouth sores that make it difficult to eat and drink. So mucositis we almost never see with an allo transplant. There is some, whenever you get chemo, I think that there is some risk of nausea. I personally don't think it's as bad as from the chemo part with the lower doses of chemo that we give compared to melphalan. There are medicines, there are immunosuppressants that you get after the transplant to make sure you don't get graft versus host disease right away and that you don't reject the marrow you just got. Those can cause some nausea, but I think those are oral, and so those are usually pretty manageable. I think with the low doses, the chance that you're going to lose your hair completely is pretty low. Like, I think it's very low. I think you can get the hair thinning, but like I said, it's amazing that, for me, having done this for about 20 years, where when I started, the only people we were transplanting were very young individuals, right, who got very sick from the procedure, to now having, people who are more advanced in age, do totally fine is an incredibly amazing difference. Like it's night and day.
That is
the way
incredible,
that the
and I don't think that word's gotten out.
Well, I think that like I said, I think that if you go to a myeloma person, right, and you say, look, I'm doing allotransplants, you'll get a checkered opinion about the procedure, and it is largely because there's this. I think most people have this memory of how horrible it was, right?
Mm-hmm.
And I think that for me and for us at Hopkins, having improved the safety, I think that now if you have something that's potentially curative, I don't think you can really turn your back on that. I think you got to at least figure out, just like with all treatments, who does it work best in, what is the best situation to do it in, what are things that, can help you along the way, what do you look out for? I think all of those things are now, trying to foster that in myeloma. But like I said, it is, if you, unfortunately or fortunately, if you talk to ask people about an allo, you get a very mixed bag.
I believe that. And with the graft versus host, there's a chronic versus acute GVHD. So do you want to talk about the differences between those two? And what you're talking about when you say it used to be 50% and now it's less than 10, that's probably the acute, not the chronic, right?
So it's actually with both. So both, it used to be about 50%. So the most people who get chronic graft versus host disease had acute graft versus host disease. Acute graft versus host disease is really sort of defined by timing. So it happens within the first probably 3 to 6 months right after the transplant. It is weird because the immune system that one receives really only attacks 3 organs in your body. It attacks your skin so you can get a rash. It can attack your liver so you can become jaundiced, or it can attack your gut so you can have nausea and diarrhea. After 6 months, right, chronic graft versus host disease can come up, and it can come up sort of at any time, and it can affect like any organ in your body. It can affect like common places that it affects. It can affect your skin again, where you get sort of tightening of the skin, something called scleroderma. You can get the attack of your tear glands, your lacrimal glands, and so your eyes are dry all the time. The same thing can happen for your salivary glands, so you have a dry mouth all the time. It can affect, things like your gut. You can attack your joints, like arthritis. You name it, it can happen. And so that what I tell people about chronic graft versus host disease is that is, as the name suggests, it's a longer-term issue, right? And many times it's controllable. You give people immunosuppression, but if you need to give them immunosuppression for a long, long period of time, then what happens is that they become very susceptible to infections, unfortunately. So that's the trade-off. So what I tell people is that getting chronic graft versus host disease is really a trade-off from your myeloma. It's like getting a different disease. And so you don't want necessarily to have this huge burden of chronic graft versus host. I think that's really a long-term issue. The acute part, I think, is an issue, but it's more of an immediate-term issue, and I think it can contribute certainly to the danger of the transplant. But it's not, like I said, it's not like this chronic issue that you have to live with day to day to day to day, for years after the transplant. So I think that there's like a little bit of a philosophical difference in approach to acute versus chronic. But, like I said, if you have really bad chronic GVH, it potentially is fully a trade-off from having myeloma.
Right. So, and I know there are different types of allogeneic transplants. So when you talk about you're using the low-dose chemo and you're reducing the graft versus host, Do all of the types of allotransplants do that? The haplo or the mini or the
Right. So using the low doses of the chemo is that's what a mini transplant is. So a full transplant is where we're going to give you 8 days of chemo. And a mini is, for us, a mini is 5 days of chemo, but it's not super high doses, and 1 day of low dose radiation. So it's 6 days of treatment, but it's at a much lower dose than the full transplant. So a mini transplant is using these lower amounts of chemo. There are differences in terms of like what the graft is that you get. So you can get, the most common one that people believe is important is if you have an HLH, what's called a fully matched donor. So this is typically a sibling, and if you think about it, these HLAs, there are like 10 of them. So if your 10 copies are the same as your donor's 10 copies, that means your immune systems are somewhat in sync, right? So that should lower the amount of graft-versus-host disease. It should lower the chance that your immune system rejects the donor's bone marrow. So in the past, for many, 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 many years, the ideal donor is someone who has a perfect match, right? That when they say perfect match, it's an HLA match. So the issue is that is fine if you are, a 10-year-old with leukemia, right? Because you probably have a sibling who is young, right? If you are 70 and you have a sibling, then it's unlikely that sibling is going to be 20, right?
Right.
And so one question is that, is there a difference on with regards to the age of the donor, right? And so there certainly are. And, one of the other ways to do it, instead of having a fully matched transplant, like if you get, half of your genes from your mom, half of your genes from your dad, that means with each parent, you share half of their what's called genotype, right? And the other is that it means that your kids share half of your phenotype, right? And so so in that way, if you said, well, okay, instead of doing everyone fully matched, maybe what we should do is think about using half-matched individuals. Well, that would allow people who are older, who probably have, teenage or adult-sized kids, right? Maybe you could use their kids as a donor, right? So normally in the United States now, if we wanted to do an allogeneic transplant, it's something like 40% of people will have a related sibling who's fully matched. Okay, that's sort of the number. If you say now, what I want to do is I don't care about them being fully matched. I just need people to be half matched. Well, that means that your parents could donate for you. It means your kids could donate for you. It means that your grandparents may be able to donate for you, or your grandchildren may be able to donate for you. It means that your cousins may be able to donate for you, your aunts, your uncles. So it ends up being that if you look at half matching, it's something like 95% of people will have a match, right?
Wow.
So it takes that matching issue out of or finding an equation a little, or finding a donor somewhat out of the equation. So the major issue using half-matched transplants is that in the past they've been associated with tremendous rates of graft-versus-host disease, and that makes sense because half of it they recognize, and then the other half they think is totally foreign, right? So there's a really high risk of GVH, and so the way that we've done our transplants, developed our transplants here at Hopkins, is that by using sort of this Cytoxan after the transplant, we found that if you have a half-matched individual versus a fully matched individual, the graft-versus-host disease rate is exactly the same. So there's no difference anymore. And so now that allows you to sort of say, look, if I compare older pairs of siblings who get transplanted versus patients and their kids who get transplanted, how do they do? So it seems like the younger your donor is, the better patients do actually across all diseases. And it's this weird thing, it's like getting, it makes sense. You get a younger, more vibrant immune system, and that is probably better than, getting immune cells and bone marrow cells that have been in a person, for decades more, right? So
yeah, that makes sense.
So for us now, like just about all of our transplants, myeloma transplants, if the patient is, let's say, over, around 50 or older, and they have a child who's a teenager or older, I would use a kid who's half matched over a sibling who's fully matched but is 50 or older. So I think that, so that changes the equation a little bit because there are many, many more people who can get transplanted. And I think you're doing better because you're using younger donors.
Yeah, I just read an article from researchers that were in Australia about different types of cancers, and they said it could be, how we're studying all these genetic mutations. It could just be the status of the immune system, which is allowing the cancer to grow. So maybe more study needs to happen on that, and maybe we need to balance things out a little bit on just the immune system status. So that makes a lot of sense, that the stronger your immune system, the better donor you're going to get.
We as humans are probably optimal, around age 20 or something, and then it's all downhill from there. So I think that the closer you are to that, the GVH rates are better, the relapse rates are lower, there's less complications with the transplant. Almost every parameter is better if you use a younger donor. So we have moved away from, are you fully matched versus not? That is the major parameter at most cancer centers. For us, it's really what is the age of donors who are available to you? And we, like I said, we've used cousins, we've used grandkids, if we have to, and so it's been, amazing also in that way where instead of now sort of you have a patient, you go and you get them typed to see what they have and what their relatives have. In the past, where many times you were like, well, unfortunately, you don't have a donor. Now, you don't even consider, that that's a possibility unless the person is, adopted or something. So I think it ends up being that also is somewhat of a game changer, allowing more access to transplantation for older folks.
Right.
Yep.
Right. That's amazing. Well, you mentioned earlier, now that you've been able to affect the safety and improve the safety, now you're thinking about how to make things better. So I don't know if you have any statistics about potential cure rates from allo or just even an idea of how effective it is, but you're using an antibody as well. So do you want to talk about that a little bit?
So we had actually looked at, using sort of this method of controlling graft versus host disease. We had just published earlier, or at the end of last year, sort of our experience looking at patients who had been transplanted using this approach, allotransplants, from 2003 to 2011. So what we wanted to do is make sure that we had enough follow-up for everybody. And in that, in that analysis, it ended up being that the probability of being alive at 10 years was about 50%, between 40 and 50%. And what we think, as far as patients being in a sustained remission over a long period of time, it ends up being about 25 to 30%. And so that is very, and in that analysis, nobody died from a complication of the transplant. and the graft versus host disease rates were like, for acute it was like 8%, and for chronic it was like about 10%. So there, what we know is that it's, when I talk to people, I say, it's probably a 1 in 3 or a 1 in 4 chance that an allo transplant is curative in myeloma. So one question is how,
odds when you start thinking about it like that.
I'm sorry?
Those are some pretty good odds when you start thinking about it like that.
Yeah, and I think the question is if it's safe, right, and you're getting something out and you have less threat of something bad happening going in, then I think it becomes something that's doable. If you said that it's 25% of people are cured, but you have a 50-50 shot of not making it 6 months, then it becomes a very different conversation and a very different equation. And so right now, the issue is that how do we try to reduce the risk of relapse? And what we found in other diseases is that if you use medicines that are not, let's say that they're not really curative in the normal setting, right? And this is not a myeloma. This would be in this one leukemia called ALL, or in this other leukemia called AML. What you can do is if you give medicines that are sort of effective at controlling the disease without a transplant, and you use those as sort of a maintenance after an allo, the patients do much, 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 much better. So in this one disease, ALL, typically if you did a transplant in a specific type of ALL called Philadelphia chromosome-positive ALL, it's about 30% of people are cured. If you give them a tyrosine kinase inhibitor like Gleevec afterwards, it goes up to like 70%. So it can really improve things. And I think part of that is because you have a totally no immune system now in place. So we're trying to do that with myeloma, and other people have thought of this. There's a national trial ongoing right now where people have an allo transplant. It's a full transplant, so it's high-dose chemotherapy. But what they get is they get Ixazomib or Ninlaro after the transplant as maintenance. And then there have been some studies looking at using Revlimid as maintenance post-allo, just like you would in the post-auto situation. So for us, we've been interested, in this idea of cancer stem cells, and we think that those are more like B cells rather than plasma cells, and so they have a different set of markers, and one of the markers is CD19. And so one idea is that can you try to get rid of those cells, because we think that those cells are responsible for the cancer growing back and for people relapsing. So can we get rid of those using a strategy? And one of the things that we identified was this antibody against CD19. CD19 is not expressed on the plasma cells, myeloma plasma cells generally, but can we give that medicine post-transplant to try and reduce the risk of relapse by getting rid of the cancer stem cells? So that's one of the allogeneic transplant trials we have here at Hopkins. It's a trial that's specifically for myeloma patients. If you have leukemia, lymphoma, there are other trials, but this is not the trial for you. It uses the same transplant we would do if we didn't do the trial, and really the trial is only adding in this antibody. You get it IV, you get it for about a year, and then we see what happens. And so the antibody itself has been used in a bunch of different diseases, including there's a study we did a number of years ago on myeloma. It's pretty well tolerated. There are not many side effects. And so we think that this would be, potentially a strategy that is safe to try to reduce the risk of relapse. I think that, like I said, there's a lot of room to improve. 1 in 4, 1 in 3 is better than 0, but it is not the majority of patients. And so I think that for us, making it safer, we don't have to study that anymore. What we should study now is how do we try to keep people in remissions and prevent them from relapsing. So I think that, when people ask, well, should I think about a transplant, an allo transplant, what are the specifics about it? One thing I will say is that people tend to do better earlier on in their therapy or in the course of their therapy rather than later on. If they've, in the past, the other problem has been that many patients will come into an allo after they've failed everything else. And I think that even there, you're asking a lot, right? For the allo to be effective. And so I think doing it earlier on, there was just a report from the Hutch looking at allo transplants. It looked like patients who did it like in the first line, after the first line of therapy, did much, much better than those who did it after a few lines of therapy. And then the other is that I think that it's important to try and have as little myeloma as possible when you start the allo transplant. And the reason is that the chemo we give you is to prepare you for the transplant is not effective against myeloma. So we're not getting rid of more myeloma by doing that. And if you think about it, we put in the other person's marrow and the other person's immune system. It takes some time for that immune system to grow and start attacking the tumor. It's probably 3 months, right? And so if you have myeloma and you have a new immune system, then it's a race. It's a race between what is going to grow the fastest. If it's your myeloma that grows the fastest, then the immune system is not going to work. So you want to be in as good a remission as possible, and it ends up being a little more difficult if you've been through multiple lines of therapy to get a full, complete remission at that point in time. So here, I've been recommending if we're gonna do it, I actually talk to people if they have, the people I would consider are people who have higher risk disease based on their cytogenetics, and we tend to talk to more people who are younger versus older about it. But I have become more age agnostic, and if someone has high risk disease based upon their cytogenetics, I will talk to them about doing an auto or an allo, and give them sort of multiple points of view or multiple choices, and then we sort of some people pick an auto, or some people pick an allo. Most people, I would say, pick autos because that's sort of the standard approach, and you don't really know as well what you're getting with an allo. But I think, like I said, I don't think it's good to wait, personally, because I think that the ability for it to work, that opportunity you get for it to work, I think really goes down if you've relapsed multiple times, unfortunately.
Yeah, I agree. I think they say that about regular stem cell transplant too.
Yep.
Like doing it earlier up front is a better approach.
Correct.
Before your disease has gotten more sophisticated.
Yes.
Well, let's talk for a minute what you mentioned before about targeting CD19 and these precursor stem cells, or what you call their B cells versus active myeloma cells. Can you explain that for patients who've never heard that before?
Sure. So if you think about what plasma cells are, like they're cells that their job is to be part of the immune system, and their job is to make immunoglobulins or antibodies, right? And so what we see as your M spike, or what we see as your light chains, your lambda light chains or kappa light chains, those are part of antibodies, or those are antibodies that are being made, and you normally need antibodies as part of protecting you from a bunch of different stuff in the world. And so those plasma cells, their normal counterpart, myeloma plasma cells are similar to normal plasma cells. And normal plasma cells, their job is to make antibodies. They're like these little tiny antibody-producing factories. Their job is actually not to make more of themselves, right? So if you want more plasma cells, it's not plasma cells making more cells. It ends up being that they come from an earlier cell. So you have a more immature cell that now matures to become a plasma cell, and that cell is called a B cell. And so if in the normal system, B cells are the things that are like giving rise to plasma cells, then it may not be a crazy idea to think that in myeloma that the same sort of system is set up where B cells are making, B cells related to the myeloma are actually making myeloma plasma cells. So we sort of looked into this a number of years ago and think that at least in some patients, the B cells are pretty important. The problem is that B cells are really different than plasma cells. They are killed by different kinds of chemo. Things that work in myeloma generally don't work so well in lymphoma, for example, that are B cells. So you have this dichotomy of the cells and what you can treat them with, and so that may be one of the reasons why many of the therapies we use are unfortunately not curative, is because we're doing a really great job getting rid of the plasma cells, but those drugs that we're using are not getting rid of sort of the factory where these things are being made. And so we have proposed this for many years, just because the allotransplantation thing, to me, is humorous because it's a very controversial thing, right? So for me, like this idea that B cells were important was a tremendously controversial thing at the time, and so for now, when I talk to people about allos and they say, well, this is crazy, blah, 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 it doesn't faze me as much because I think I went through the whole skepticism with B cells way back when. But so, one question is that how do we know that B cells are important? And actually, it wasn't my group or our investigators at Hopkins that provided evidence. It was actually a group at the University of Pennsylvania. So what they did was they had these CAR T cells, right? They were one of the first groups to generate CAR T cells. So they had CAR T cells, but not against a myeloma protein. They were against B cells. They were against CD19. So there are a couple of people up there. One is Carl June, the other is Ed Stoutmeyer, and Dr. Garfal. They had a trial where they used those T cells, so CD19 T cells, in patients with myeloma, and in some of the patients they saw, pretty good effects of getting rid of, tumor, even though CD19 was not on the tumor cells. So that was a study that was published a couple of years ago. That, too, is highly controversial if you ask people. But I think based upon that, one question is can we design, CAR T-cell therapies that, let's say, there are CAR T-cell therapies now that go after myeloma cells. Those are against a target called BCMA. So one thought is, can you combine a BCMA targeting T-cell to get rid of the myeloma with a CD19 targeting T-cell to try to get rid of the, the B cells? And so that's a study that people are thinking about doing, in a couple different places. One for sure is up at the University of Pennsylvania. And so I think that there are, for me, I think that there's potentially some utility in doing this. I think that in the post-allo transplantation is probably the best time to use any immunotherapy, whether it be CAR T cells, whether it be antibodies, because the immune system is just much, much better than the immune system in the patient was, because that was hampered by myeloma. And so I think that, doing this trial where we give this antibody against CD19, in my own mind, is sort of a value-added thing where I don't think there's going to be side effects from it. Of course, we have to do it under a clinical trial to figure out whether there are. But given that it's been pretty safe in patients just who haven't had a transplant, I think that there's hopefully some benefit to that in terms of preventing relapse without adding a whole lot in terms of side effects and toxicity. So that's sort of the rationale for the trial we have right now.
Well, that sounds great. I think it'll be so fascinating to see. And what stage is it in? What phase trial
So
is it in?
it's a phase 2 trial. It is one that is sponsored by in part by the NIH, and it's in part sponsored by the company that makes the antibody, MedImmune, which is AstraZeneca now. And we actually just we just finished our first patient, and we have a couple more that we're getting ready to do allos in, and then we're signing them up for this trial. So it is, I think it's an ongoing effort. I think that, it's interesting. One thing that's interesting to me is that, really the push to do more allos here has been over the last, I would say, 2 years maybe. And, in the beginning, there were really no patients that we would consider doing allos in, but seeing that they're relatively safe, it doesn't, if it doesn't work, you can still go on to other therapies. So over the last, I would say, probably year and a half, I've gotten a lot of referrals specifically for allos. And so I think it's nice to see that some people are interested in at least, finding out whether or not that might be an approach that they could think of.
Yeah, well, that's wonderful, and I think it's a great option for people. So I think it's like I said. I think it's just having a conversation with someone about it. It doesn't mean that you go and you talk to someone about it that's what you're going to get. I think you have to go into the details and figure out is it for you or not, right? Are you the right person to do it in? If you had low-risk disease, I would say, look, there are better things out there, and we may, not even need allotransplantation. We may have curative things in 10 years, and if you have low-risk disease, you probably make it that long. So I think it really is a pretty involved conversation that's very, very specific to an individual patient. Yeah, right. Well, one thing that you said, I want to ask one more question before I open it up for caller questions. When you talked about using drugs that were for the B cells, like the CD19 target or other types of B cell cancers, have a lot of different myeloma specialists tried to combine kind of the standard care therapy in myeloma with something like that to besides this antibody that you're using?
So
to see if that even works?
So we had done a trial using a different antibody called rituximab, which is very commonly used for lymphoma, and we did that almost a decade ago probably, and we didn't really see anything, right? And I think that we sort of understood some reasons over the years why that we think that that's the case, and I think that this antibody is better than that antibody. It's a controversial enough thing, and the other thing is that most myeloma people are not like lymphoma people, right? And so most myeloma people are pretty specialized, and so I think that the ideas of, like, many of the diseases, like I said, the therapies are the same, right? Like, we know how to use Velcade, we know how to use IMiDs. When other people try to use them in other diseases, it's a bit of a head scratcher for them. And so I think that the comfort with combining, those types of strategies, I think for sure, number one, has to be done on a clinical trial, and number two, I don't know if that really hit the radar of a lot of people. Like I said, we have our data, and it is super controversial still. And I think that, but that's what science is for, is to sort of figure out and
try things out.
These things.
Yeah.
And figure out what is really going on. And so, but I think that that's sort of what I would say, is that I think some people are pretty open-minded, and other people just say, look, forget it. That is completely made up and false, and let's not even think about that. So, like I said, it's science, and I think if you do things under the right conditions, like doing them in a clinical trial, you don't just make them up on your own. I think that that's really and you explain what is going on to the people who are enrolled on the trial, then I think that's probably the best situation to try and figure actually these things out.
Yeah, wonderful. Well, let me open it up for caller questions. I still have more questions but I don't know how much how much more time you have. So
okay.
Let's just go ahead with caller questions. If you'd like to ask Dr. Matsui a question, you can call 347-637-2631 and push 1 on your keypad. And we'll start with our first caller, 9836757. Go ahead.
Hi, good afternoon, Jenny and Dr. Matsui. This is Dana Holmes. Thanks so much for this interview today. The information that you're sharing today is just so fascinating. Just to clarify, is this trial that you're speaking of, the allo trial with the CD19 targeted antibody, is it for newly diagnosed high-risk patients or is it for relapsed and refractory patients?
So it's actually for both. And so for newly diagnosed high-risk patients, it's based upon your either your cytogenetics or your gene expression profiling, right? And if that's something that's still done, and so for newly diagnosed patients, that's the thing. And people, like I said, need to go into a good remission with their initial chemotherapy. As far as relapsed patients go, it is open for relapsed individuals. The population we had initially been focusing on were patients who relapsed pretty quickly after an auto. So I think that if there's relapse that occurs within 18 months of having the auto, historically that's been a tough group of patients to treat long term, and so we focused on those. But like I said, I think that the major, over the last year and a half, I think the important lesson that we've learned is that you really need to be in a very good remission, if a CR if possible, going into the transplant, because you really got to give that new immune system a fighting chance and time to grow.
So an allo transplant at initial diagnosis is actually a practical consideration based upon this trial design.
It is, but, like I said, if you ask, 100 myeloma experts, right, and you said, look, I have a young patient that has, the worst of the worst, they have a 17P or something, really bad, what would you do? I think some people would consider an allo transplant at that point, but the vast majority would not. They would say to go onto an auto transplant and maintenance. Patients are eligible, and like I said, I try to have as balanced a conversation as possible, right? These are the standard approaches. This is what auto transplant entails. This is what you probably get out of an auto transplant, and this is the potential of an allo, and try to help the person, think about it and make some decisions that way. But for sure, people say, well, if I send them to you, you're just going to do an allo transplant. I say, no, come on. Part of talking to people is explaining what the different options are. And so it's not like the vast majority of people who come to see me for,
I don't think you're going to slap the handcuffs on them and tell them you can't leave until you get the allo.
The majority of people still vote to do an auto transplant. And I think that that's fine. As long as they feel comfortable with that decision, as long as they feel they got the right information.
It's obviously standard of care, and everybody pretty much leans towards what we know in standard of care. But I think in the back of my mind, I always think, well, gee, standard of care is not curing anybody, so why not take the...
I am totally with you, and so that's sort of the approach we take. And like I said, I think that we've done a lot here to make transplantation safer, and so I think that is one argument that, in the past was a very valid one, and I think that is actually not that valid an argument anymore, that it is much, much more toxic than an auto.
And I understand and respect the fact that studies need to be completed in order to validate these premises, but I thank you for pushing the envelope, because it just may end up being something that we all need in the myeloma community. Dr. Matsui, I have a question, though, because it always tends to puzzle me, and I think I kind of fall back to the fact that this disease is so heterogeneously distributed throughout the marrow bone marrow bias is what I generally refer to it as. And the sample and corresponding results are only as good as the sample procured. So if you're going to kind of be considering whether you're high risk or standard risk, how does the bone marrow sample bias affect the determination of the actual cells, the disease phenotype at any given point, whether it's a diagnosis, at relapse, using the MRD testing? Can you help me better understand that? Is there a way to overcome that?
Right. So there is a lot of what's called intratumoral heterogeneity. So in each patient, there are, like, if you sort of said, look, this myeloma has this number of mutations or these specific mutations, and you could have other myeloma cells that have a different, for sure some shared mutations, but a variable other ones, and so that's where the heterogeneity comes in. And I think that the other thing you can see is the your genetics or cytogenetics changing over time. And that's probably an evidence of heterogeneity. So this issue of sampling is a real issue. Nobody wants to get like 6 different bone marrows to sample 6 different places. But I think that what I do is you got to go with what you have, and we know that certain cytogenetic abnormalities are bad from doing a single marrow. So I think that part is okay. Typically, what I think about is if patients are not really responding well, especially to their induction therapy, I like to remarrow them just to make sure that there's no something else that we didn't catch the first time. So I think that, like I said, there's no perfect strategy.
Because that would make sense. If you found the high-risk markers initially, you're kind of okay with the choice. But when you don't, and you're being told that you're standard risk, and you're kind of going that standard risk, standard of care approach, that's a scary thought because
and I think that if if you're standard risk, then your myeloma should act in a standard way, right? And if it's not acting in that way, I would actually go back and try to refigure it out.
Good
there's
approach.
Something weird, right? And so if there's something weird, then I don't think you can just sort of blindly say, look, this is low-risk disease, and so, you got low-risk disease. I think that, low-risk disease responds well to chemo, and so I think that if you're not seeing responses, then perhaps rechecking would not be a bad idea.
Does the aspirate sample need to be the first pull for the MRD testing or any type of testing? And if yes, is there consensus from center to center doing these types of tests?
No, I think that for most tests, I don't think that the first pull is always done. I think that there is issues, depending on. I think if you do 2 pulls, I think the second pull is probably as good as the first pull. I think that if you're doing, 5 pulls, I think the 5th pull is probably not very useful. But I think that at least a couple pulls, I think it's okay. I think that there are pretty standard ways that we do it, the order in which we get marrow for tests and what tests we, send off first, or what samples we use for each test. And I think that's very similar in many different centers. And so I think as long as it's uniformly done, I think that you can, at least have confidence in that. But is it a perfect system? For sure, it's not. It's not. Difficult. Everyone knows it's difficult doing marrows. Sometimes it's like, cake, and other times it's like really difficult, and there's no explanation for it. So I think in those ways, it is a little bit of a skittish way of doing things.
Well, thank you so very much for your time today. I wish you the best of luck, and hoping that maybe someday the Livestrong Center will also open up a satellite on the East Coast again so that we can get you back on the East Coast as well. So good luck. And Jenny, thanks so much for taking my calls.
Thanks, Dana, for your questions. Good questions. So we have another call, 412-9050, and then we will end with this question. Go ahead with your question. Hello? Well, we will try 310-5598. go ahead with your question.
Hi, Dr. Matsui. So I don't want to take a lot of your time. I know we're just about done with the call, but I just want to thank you for everything you've shared. It's been so helpful. My question is regarding the different cancers you've worked with. So it looks like you haven't solely worked with myeloma, but you branched out to other cancers as well. So what I'm wondering is, what lessons have you learned while working with myeloma in comparison to the other cancers you've worked with, and also how your experience with other cancers and working with these cancers impacts myeloma?
So I think that, and I think that, so we've worked on leukemias, and the other thing that we've worked on is pancreatic cancer, and we've done the same sort of thing, looking for cancer stem cells in all of those diseases. Really, the reason why, people always ask, well, why are you doing this? So the reason why is because I train a lot of people, and if people have specific interests, I want them to follow those interests. I don't want to force a leukemia person to become a myeloma person. So that's how they've sort of developed. I think that, in some cancers, it's really well understood, like leukemia, where stem cells are, how they're related to other cells. So I think that is enormously useful as a roadmap for us to study myeloma. And that work has been going on in leukemia for much longer than, our work in myeloma. And I think in pancreatic cancer, the one thing is that it is, I would say, a little bit easier to study the environment, right? Like, I think that we know the environment is important in myeloma, but how do you study it? Because it's inside of a bone, you pull it out and it's a bunch of liquid. And so in solid tumors, you get chunks of tumor and you can sort of see what cell is next to what and what are they doing. And so I think that from pancreatic cancer, we've learned a tremendous amount of how do things in the environment regulate cancer stem cells in pancreatic cancer. And so I think that is very helpful for us to now start thinking about, well, what are the things that really control these cells in myeloma? So it's been having, different ways to get at a problem, I think is incredibly helpful, right? It hopefully prevents you a little bit from getting tunnel vision and thinking that everything you do is right. But so I think that we've learned a lot from those other, my lab has learned a lot from studying those other diseases. For me, clinically, though, I only see myeloma patients. Like I am a transplanter doing other diseases, but, all of my time in the clinic is seeing, folks with myeloma and other plasma cell diseases. So it's my first interest, my first, the thing that I'm for sure most interested in, but I think that the other things are, like I said, very helpful.
Yeah, that's great. Thank you so much.
Okay,
sure.
Thank you so much. Well, Dr. Matsui, thank you so much for joining us. I think your work is really exciting, and I hope you stay open-minded like that.
Oh, yeah, that won't be,
those maverick
hopefully.
Things, yeah.
Yeah, I don't think that'll be that difficult. So, well, thank you so much for having me.
Well, we're just really appreciative for all you're doing, and we encourage people to attend the roundtable, also to meet you in person and ask you questions about their type of myeloma and other things like that I know you'll talk about there, and also to visit you in Austin when you get there.
Yes.
So we're very excited for you. We're just thrilled that you'll be leading that program.
Yes, I'm excited for myself, too.
And you'll like the weather a lot better, probably.
Yeah, I think that'll be nice.
Okay, well, thank you so much again for joining us. And
Great.
We'd like to thank our listeners for listening to Myeloma Crowd Radio and invite you to tune in next time to learn more about the latest in myeloma research and what it means for you.
Lucky Land Casino, asking people, what's the weirdest place you've gotten lucky?
Lucky? In line at the deli, I guess?
Aha, in my dentist's office.
More than once, actually.
Do I have to say?
Yes, you do.
In the car before my kids' PTA meeting.
Really?
Yes.
Excuse me, what's the weirdest place you've gotten lucky?
I never win and tell.
Well, there you have it. You can get lucky anywhere, playing at LuckyLandSlots.com. Play for free right now. Are you feeling lucky? No purchase necessary. Void where prohibited by law. 18+. Terms and conditions apply. See website for details.